Good evening, everyone, and thank you for joining us for the Snowdome Foundation 2021 Blood, Sweat and Tears of Joy educational event. Let me begin by acknowledging the Wurundjeri people of the Kulin Nation, who are the traditional custodians of the land of which we gather, and may we pay our respects to their elders, past and emerging, and to other Indigenous Australians here today. We would have loved to have seen you all in person, but this year seems to be as unpredictable as the last. However, on a bright note, Zoom has allowed us to expand virtually across Australia. So on behalf of our chair, John Salvaris, the Snowdome founders, the board and the executive, I'd like to extend a very warm welcome to the more than 380 people joining us online tonight. Not only is today the first day of spring, I hope people got to enjoy it. It was stunning in Melbourne. September 1 also marks the beginning of Blood Cancer Awareness Month. And it pains me to say that when combined, blood cancers are now the second most diagnosed cancers and one of the highest causes of cancer death in Australia. It's claiming more lives than breast cancer and melanoma combined. And in addition, the incidence of blood cancer continues to grow. Over the past 10 years, blood cancers have increased by 40%. And when you look at in perspective of numbers, in 2021 alone, more than 18,000 Australians will be newly diagnosed. And that's the equivalent of 50 people every day or one person every 28 minutes. There are no screening programs available and no way to prevent blood cancer through lifestyle change. So what we need is greater awareness and a focus on funding translational research to find a cure. So this year, Snowdome is asking you to get involved by kissing a vice goodbye for the month of September and then donating the savings to help fund more blood cancer research. You can head to our website to learn more. You'll also be able to see some of our founders and some of our board and our execs and the advice and the advices that we've given up. And if you care to make a suggestion, Miles is yet to figure out one, so I think you could use some help. But we do encourage you to go on after the webinar, have a look around. You can see our new portal, and the website is www.snowdome.org.au. Before I introduce our host this evening, we've got a little housekeeping to note. We would encourage you to ask questions throughout the event, and you'll be able to do this by submitting your questions using the Q&A button that you'll either see at the top or the bottom of your screen, depending on what device you're using. As I said before, there are 380 registrations, or there's more than 380 registrations tonight. So we'll endeavour to answer all the questions if possible, but we may not get around to all of them, but keep them coming because we can answer them at a later stage. I'd also like to take a quick moment to acknowledge Vince Giarusso, who created the beautiful music playing as you join this evening. The piece is entitled Stay and Wait and is in memory of Chloe Rutherford. Now that we're all set, it's time to sit back, get comfortable, pour yourself a glass of wine, and allow me to introduce you to our host for this evening. I'm sure he doesn't need any introduction, but as you know, Professor Miles Prince is an internationally recognised blood specialist and Professor of Medicine at both Melbourne and Monash universities. He divides his time between Epworth Hospitals and the Peter MacCallum Cancer Centre. Miles is a very active clinician, overseeing clinical and laboratory research, especially in blood cancers. He is a co-founder of the Snowdome Foundation. He also founded the CAR-T program at Peter Mac more than 15 years ago, and in March of this year opened the Epworth Centre of Immunotherapies and Snowdome Laboratories. Miles is a fellow of the Academy of Health and Medical Sciences, and in 2014 he was appointed a member of the Order of Australia for distinguished services to medicine, particularly in the areas of blood cancer research, patient care and philanthropy leadership. Ladies and gentlemen, Professor Miles Prince.
Thanks so much, Kirsty, and thank you everybody for joining us. We've got a huge crowd, and I know it's going to be a really exciting next hour and 15 minutes. We plan to finish at 7:30, and the way we've sort of worked this evening is that I'm going to give a short presentation, and really it's all about hearing the stories of our panel members. And so we've got some really amazing people to join and discuss their journey in different ways through blood cancers. I'm going to introduce them in turn, but you can see them. We have Janet Gregory and Deborah Sims and Barry Bois, all patients and who lead amazing lives in themselves. And we have Sam Fiorenza, who is a specialist haematologist who is focusing on his work towards new treatments for patients with blood cancers. So who could be better to join us? And one of the keys and passions of Sam is CAR T cells, which we're going to really focus on tonight. So a lot of you may or may not be aware about CAR T cells. So I thought what I'd do is take the opportunity to give you a short presentation. I'm just going to spend a few minutes really talking about this concept. So we know in the past that there has always been three major pillars to treat blood cancer, which we still use nowadays, and that's surgery to remove tumours, not so much used in blood cancers, but certainly for diagnosis. Radiation treatment is very critical, and what we call traditional chemotherapy. And there's been two new pillars that have been added to that. One is called precision therapy, which is matching the right drug to the right patient. And Deborah Sims is going to talk a little bit about her experience, because she's had some drugs that we would consider precision therapy, very focused treatment, which is not chemotherapy, but new drugs. And she's had those in the context of clinical trials. And then this concept of using the body's immune system to kill cancers. Now, only 500 million years ago, we decided that we were going to evolve from jellyfish. And strangely enough, that was an important step as we discovered that the immune system was incredibly important. A jellyfish basically recognizes any sort of insult, whether that's something standing on them, sand, something trying to attack them, or potentially poisons, as being foreign. They're all the same. A virus, if it gets attacked by some sort of bacteria or virus that might be in the water, it will see it as the same as if it was being insulted by sand being thrown in its face. However, sea lamprey, which they're pretty ugly looking things, but they're called jawed fish. And they are the first, and they still exist in northern Scotland particularly, and they have lymphocytes. They have an immune system that is able to recognize what is self and what is not self. And that's the fundamental aspect of our immune system, the capacity to recognize what is foreign, what is not us. And the cornerstone of these are white blood cells called lymphocytes. So in our blood, we have lots of different white blood cells. We have neutrophils, lymphocytes, basophils, monocytes. They all kill. That's their job. They're like the military, they kill. But like the military, there's an army, an air force, a navy that kill in different ways. And one of the key cells are the lymphocytes, and of a particular type called a T cell. So this is a T lymphocyte. And what it does, it has an attack on a foreign cell. And that cell can be a virus, or it can be a bacteria, or it can be cancer. And it binds on and it forms this kiss of death. What happened in 2018 was a Nobel Prize winner, Honyo and Allison, recognized that our immune system is fundamentally switched down. It's suppressed. So what happens is, as soon as cancers grow, one of the first responses that cancers have is to suppress the body's immune system to attack them. So as our body is suppressed to stop us getting autoimmune diseases, because if it wasn't, we'd get rheumatoid arthritis and lupus and multiple sclerosis. So our immune system is suppressed. And that was what was discovered by Honyo and Allison those many years ago, awarded the Nobel Prize. But what happens with cancers is that they subvert the immune system and push it down further, so that our body won't recognize and kill the immune system. And so that's part of the reason that these cancers grow. So what we have discovered is that we can actually modify T cells to better recognize the cancer. This is the cancer cell which we want to attack. And in this situation, it's called a B cell. And it can be an acute leukemia cell or lymphoma. And these T cells are being pushed away by the immune system. They won't recognize it. So what we can do is modify that by gene therapy, by putting specific genes, which if you amplify this, you see this process here, where this new gene forms this hook, which is able to then hook onto the component on the surface of the cell called CD19. So this is like a Sputnik binding. And so this pulls it together. And so we've got the good guys who are the T cells, and we got the bad guys. And the whole concept is to bring the two together. And so I'll just show you some work that we did in our lab, which was what we call these sequential killing, or what we call multi-killers. And so this, I'm going to show you a target cell, which is otherwise called a lymphocyte, and it's going to be attacked by the CAR T cells. So it should come on. So this is the cell that is the bad guy, and along comes a CAR T, which is this modified cell. It binds on, it injects it, it kills it. And that's the process that we see where it physically binds and kills. What actually then happens is a bit more detail, because these are serial killers. The individual CAR T has the capacity to kill not just one cell, but can kill 5 to 10 cells. And you'll see this CAR T kills this one, binds off, kills this one, binds off, will kill another one. And so I'll just show you that again. You can see the binding, it kills it, binding, it kills it. So these CAR Ts have the amazing capacity to kill multiple cells. So how do we do it? Well, you have the patient, and they undergo a apheresis, the standard sort of cell collection process on the machine. We then collect these cells in a bag. We separate out the T cells. The T cells are then cultured in a cocktail for about 2 weeks, where we add in special herbs and spices, and then into that to make these T cell grow. Then we add in a virus, which is actually defunct of any activity. It can't actually replicate. But what this virus can carry, like a Trojan horse, is the genetic material that allows the expression of these receptors on the surface of these T cells. So they move from these blank, dumb T cells, which are being suppressed, to these very active T cells, which are then put back into a bag and reinfused into the patient like a blood transfusion, and the result is killing of the cells. And we'll hear about the success story of that from Sam in terms of the biology, and the success story of that in terms of a patient, Deborah. So this all started with the savior of a patient called Emma Whitehead, who had an acute lymphoblastic leukemia, and this happened all back in the early 2000s. Emma had failed a bone marrow transplant. She's now cured of her leukemia. This is called CAR T cells because it's a chimeric antigen receptor. So this little protein that I showed you on the surface are called antigen receptors it binds onto, and it's a T cell, and it's a living drug. It can cure acute leukemia. It's now used in lymphoma and myeloma, and we will talk about that a little bit later. It may well replace bone marrow transplants. It's being used in other blood cancers, which we'll talk about with Sam. It's a one-off treatment. It's expensive. It's now approved in Australia for a variety of indications, and we'll talk about it. But it's not without its side effects and a toxicity. And so anybody who undergoes CAR T cells, it sounds simple, but I bet you Deborah was pretty scared when she had those cells infused. What is my immune system going to do to me? And we'll touch on that. But the elephant in the room for this example, called Chimera, is that this in US dollars is not cheap at the time, and it's still a very expensive treatment, but it's a one-off therapy. And again, Sam is going to talk to us about this elephant in the room. So it's footy finals. Tigers are out. Things do change. This is 2019. But this was a publication of where we are in Melbourne, the city of hope, because there was a big allocation of funds from the government to grow CAR T cells in Melbourne, specifically at the Peter Mac. And it is a major breakthrough in cancer. And it is reason to be excited. But one of the things that we need to do, and one of the things that Snowdome is focused on, is getting more CAR T cell therapies around Australia. That is the end of my presentation, and now I really would like to talk to and with everybody. The focus of the evening really is about both clinical trials giving us hope and the new therapies, and we chose CAR T cells as being one of those. And I'd like to go through introducing each of our panel members and really talk about what their story is and how their journey with blood cancers has evolved over time. And all of them have been over the last decade. These are not short stories. And that's what makes it so exciting. I'll be blunt with you. If you look at the stories, and if we were 20 years ago, we wouldn't have the panel together here. That's how different it was. These people are alive because of the evolution of cancers, and we're lucky that they've joined us today. Janet, I'm going to do a quick introduction, and we need to take you off mute. So Janet Gregory is a patient of mine, and I know Janet very, very well over the last few years. She's an amazing person, and I really want you to hear her story, and I'm going to talk her through that story over the next few minutes, and then talk about what has evolved with Janet and how she's been saved by clinical trials. So Janet, thanks for coming. Janet's sitting at home. Janet, you'll understand that Janet has some problems with her speech, which is a result of the complications of some of the treatment that she's had along the way. She speaks beautifully now. At one point, she couldn't speak at all. But Janet has a really interesting past. So Janet, when all of your problems started, you were in Borneo.
Yeah.
How were you there? What happened?
Well, I took a contract as a CEO and Deputy Vice Chancellor for Swinburne University, which is an Australian university in Melbourne. So I went up there originally for 3 years with my husband, who liked the idea of playing golf and things like that. And it was a fascinating but also very demanding job. So I think when I first got tired, and I mean really tired, that was probably about 6 months before I was diagnosed. But typical me, I put that down to work. And it wasn't until I got a rash. So this is all in 1917. And I got the rash in July 1917. I flew back to Melbourne for work. The rash went, which made me think it was a topical rash from Borneo or something. It went, but when I came back, it came back again. And I got tireder. The rash didn't really go. I went to the local, very small hospital, and they put me on steroids for lupus. They thought it was lupus. So the steroids probably didn't help. And eventually they said, leave hospital and go to a resort to relax. But I decided straight from the airport to Epworth Emergency. My adult daughter said I looked like a pink chipmunk because I was so bloated and red.
So Janet, at some point they did a kidney biopsy on you, is that correct?
Oh yeah, that was in Borneo.
Great place to have a kidney biopsy.
I think in retrospect, bad decision, because I think it introduced bacteria.
Yeah. So
it wasn't sterile.
So you were diagnosed with this T-cell lymphoma.
But not until November. So at Epworth, they thought it was a tropical disease and kept treating me for other things, and eventually called in a hematologist who said you need a PET scan immediately. And I did that and lit up like a Christmas tree. So that was cut weekend, and my sister was asking medical friends, so we asked you to come in and talk to me as the best in the field for T-cell lymphoma. Yeah.
So I'll tell everyone your story because it's very interesting and complicated. But basically, Janet had very, very bad disease, and it was throughout her body. And she had a very rare type of T-cell lymphoma. And T-cell lymphomas make up about 5 to 10% of lymphomas. And unfortunately, most of them are not curable. And a lot of them, in fact, only about 30 to 40% respond to frontline treatment. So Janet went through aggressive treatment, chemotherapy, and really went through it extremely well. And the PET scan improved and things were looking really good. And in fact, we were heading towards a stem cell transplant. But what happened was that she then developed a bad infection and a fungal infection with a particular fungus called Scedosporium. The survival rate for Scedosporium is less than 5%. And we actually found it in the kidney. So we, Janet, after much discussion, said, okay, I'm prepared to lose my kidney. And we removed part of the lower part of her muscle and skin on her abdomen. And she was saved of the Scedosporium, never to be seen again, which was actually quite remarkable. And was doing fine, in remission, but then one night problems occurred. Can you tell us what happened then, Janet?
Well, I had what I believe is a clot in the lung that bled profusely. And at the same time, I had cardiac failure. Luckily, I may be using the wrong terms, but luckily a nurse was in the room, so pressed code blue, and ICU team was on the floor. So they rushed down, kept me alive. I think I was almost bedridden, and rushed me to ICU, and I spent about 3 weeks in ICU with early diagnosis being that I wouldn't survive.
So
But
I'll just interrupt you there, Janet. So basically, I think Peter, who you saw pop in before, behind you, Janet, he was about to sneak up on you, but Peter sat there for 3 weeks while we waited for you to wake up. And Janet woke up and she had this very rare condition called Lowndes-Adams syndrome, which is, I think there's about 5 reported cases, and it affects a certain part of the brain where you basically cannot talk, and you have this severe tremor and talking and eating, to the point where Janet could not talk for weeks. And then along the way, Janet, not soon after, things started to recover, but because of the delay in all of the treatment, your lymphoma returned.
Yeah.
How did you feel?
Devastated, I guess, because I was in rehab by then and trying to get some movement back. I couldn't walk, I couldn't eat, I couldn't really do anything by myself. But the cancer meant, or I thought it meant I had very limited time. So what's the point in doing all that rehab?
So
that's what it felt like.
We had a fairly blunt discussion, and we talked about the fact that the cure rate with relapsed lymphoma is extremely low.
Yeah.
What year are we talking about these? 2000 and...
I think 2018, I think.
Yeah, 2018. So, you know, a year had gone by. And the survival of relapsed T-cell lymphoma is usually less than 6 months. Most people do not survive 6 months. So we didn't have a choice. Janet had no option for a transplant. She was in bed. She was basically bedbound. She couldn't walk. So she was able to get this drug, which had been funded by Snowdome as part of a clinical access and research scheme to look at the biology of T-cell lymphoma. And this drug's called romidepsin, which is not routinely available in Australia, but is available in the United States as the only part of the world. And we gave her romidepsin, which is given as an infusion once a week, 3 weeks on, 1 week off. And that was, how long ago did you have your first dose?
3.5. Oh, I'm so bad at remembering.
Yeah, but it's about
2019, maybe. I think.
Yeah.
And
so we're approaching nearly 3 years. Yeah.
I'm now on 3 weekly doses. It was 2 weekly and a 2 weekly alternate transfusion.
And in terms of your rehab?
It's going okay. I'm walking with a frame, but I'm walking. So that's pretty exciting.
So Janet, I'm going to come back to your story at the end. Sam, I'm going to ask you, before we talk about your story, as a medico, clinical trials, it's all about discovering, new ways of treating things. But are there other advantages of clinical trials?
Yeah, thanks, Miles, and thank you, Janet. That was an incredible story, actually. It was an amazing story. So the other great advantage of a clinical trial, Miles, is exactly what you were alluding to before with the romidepsin trial that Janet was on, which is getting some correlative biology and pathobiology out from these trials to understand about how to improve these therapies better. And that is key. You know, a clinical trial is only as good as the drug you put in, but is made even better by the information you get out. And I'm always enthused to read clinical trials, not just to see their results, but to see how we can make the next thing better.
So Deborah, I'm going to introduce you very briefly because I'd like you to introduce yourself because I wouldn't be able to do it well. But Deborah, you have been, you've been a very active in life in many aspects, and you were diagnosed with your condition about 10 years ago. Can you tell us a little bit about yourself before you got to that, and then take it from there?
Yeah, thank you, Miles. And it's a huge honour to be here. And Janet, your story has really made me quite emotional. I was a 38-year-old mum of 3. I'd been a BBC journalist for 12 years and had moved to Australia with my family. And I was feeling tired. I'd had a little lump in my neck, which I had been to a doctor to get checked out, and I'd had pneumonia 4 months earlier. And the doctor did the right thing, sent me for an ultrasound and a blood test. Nothing showed up. And so I, thought, well, fantastic, don't need to worry. But he did say, if it hasn't gone in 6 weeks, come back. And 6 months later, I went back to my doctor with my 2-year-old daughter who had an ear infection, and it was a different doctor. And she said, Deborah, I haven't seen you in so long. I'll look at your medical records while you're here. And she found that I'd had this reporting of a lump, and she said, oh, have you still got that lump? And I said, yeah, and there's a few others. I think I'm allergic to the guinea pig. And she said, okay, have you got insurance? Because I want you to see Miles Prince today. And she then made me leave the room and rang Miles. Miles was at an international medical conference. So I saw someone else in Miles's practice, Melissa Keneally, who was fantastic. I went from my doctor's literally with my daughter to Cabrini. And then it was, have you eaten today? Because we actually need to do a biopsy. And I had a lymph node biopsy on the day, that I'd reported this lump in my neck.
So Deborah, you were only 38.
I was only 38. And so my doctor said to me, and she hugged me before she sent me off to Cabrini. And that's, if your GP's hugging you, that makes you really worry.
Yeah.
She said to me later that she assumed I had lymphoma. So I went and saw Melissa, and 4 days later, I go back to Cabrini, and the minute she comes out, I know it's not good news. And I've got my daughter with me and my husband. And Melissa says to me, I'm very sorry to tell you, the biopsy's come back. You've got an incurable form of leukemia, and it's called chronic lymphocytic leukemia. And the average patient is a 72-year-old man. You're a 38-year-old woman. This is very unusual. What's more, you have a subtype of CLL called SLL, which again is rarer still. But I can assure you that we are going to refer you to everyone you need to see, and you're going to have every test you need to see, and we're going to find a solution for you. But the standard treatment for CLL at diagnosis is no treatment. So for any CLLers on here, you certainly know how hard that is. So I got put into a period of watch and wait, or watch and worry, as we patients like to do it. And because I'm a journalist, I did this sort of out of body experience where I treated it as a journalistic exercise. So even though it was happening to me, for the first 2 weeks, it was a week before Christmas. I've got 3 children. It's I'm told when we get further investigation into my markers and the CT scan and my I didn't have a genetic test at that point, but I did have a FISH test, which showed that I did have quite an aggressive form of the disease. And at that point,
so Deborah, so the FISH is not something that's swimming or a thing.
No, but it is a bit fishing with probes to find out. And I had a rare, again, lucky me, a rare subtype, which showed I had the deletion 6q. And that wasn't something that, but it was an intermediate marker for aggressive disease. But my neck was blowing up. My blood work was fine, but all my lymph nodes in my body were growing quite quickly. And the decision was made to put me through to chemotherapy because in 2011, that's all we had. And I, at this point, using my journalistic brain, had jumped onto forums in the US, learned about clinical trials, as many patients here probably have. And, you're doing all the middle of the night. I'd started mourning myself really quickly, and a friend who actually had sarcoidosis, the best advice she gave me after being newly diagnosed was, don't mourn yourself too quickly.
So you mean that feeling of just saying, I'm going to die?
I'm going to die. I'm going to leave my children. I spent that Christmas, I took about 5,000 photos that Christmas. I did a cookery book. I did a cookery book of all the family recipes for my children to inherit. And this was like in the first month after diagnosis with a chronic leukemia. But I just did Dr. Google, which Melita had told me not to do Dr. Google, but I did Dr. Google, and the stats weren't good. I could not find any younger patients that had lived beyond that 5 to 7 year period. And I fought against having chemo, and I got second opinions, and I really didn't want to have chemotherapy. There were these drugs in trials. There was something called PCI 37625, and then there was something called ABT 199, and surely I could get these drugs. Why can't I go on a trial? Why do I have to have chemotherapy? Because as far as I was concerned at that point, everyone who had chemo, the minute they relapsed, it was speeding up. The clock was ticking quicker. And I was told that I may not get a good remission from the chemotherapy with the type of disease I had. But I'll never forget, because I did manage to hold off for a year to have the chemotherapy. And Melita said, look, okay, have one more Christmas without it. But I'm telling you now, you're going to be really sick and you're going to regret not having the chemotherapy. And I started chemo on January the 3rd. January the 6th, I could turn my head for the first time in months. And I was like, why didn't you tell me to have this before? I mean, I feel so much better. And I did. Chemotherapy saved my life. And I had 3 cycles of FCR. The plan was I was referred immediately for stem cell transplant. So the plan was, as soon as I relapsed from FCR, as they expected me to, as from the chemo, I would go straight to transplant. So I laugh when I see David Ritchie, because we're 10 years on now, but every time I saw David Ritchie at the Royal Melbourne, who's a prospective, he was my prospective transplanter, and long may he stay that way. He would basically tell me all the ways that things could go wrong with transplant. So I was very, if I was against having chemo, I was even more against having transplant. But got through the chemo, started relapsing. They said to me, well, we're hoping you'll get 2 to 5 years. If you do well, you'll get 5 years. I got 6 months before I started relapsing after quite serious chemotherapy. And that showed that I was in then a lot of trouble. We kept an eye on me, but at this point as a journalist, I'm going to America. I'm going to find a clinical trial. There is, Ron Walker was a friend of mine, and Ron Walker, bless his soul, went public. I didn't even know what he'd gone through. And I was reading the AFR magazine one weekend, and there was Ron, his before and after scans, having gone to California and got on a trial of a drug called Keytruda, which now thankfully we have listed on the PBS for melanoma. And because he went public, I thought, okay, well, I'm not Ron Walker, but why can't I there must be something. My children deserve a mother. They deserve me to be here. And at that point, I took out my superannuation. No one had told me that you could draw down your superannuation to help you with emergency healthcare costs. And I emailed the top CLL expert in the world at the time, and Professor Tom Kipps. I sent him an email. I said, look, I'm just a mum from Australia, and I've got 3 children. I just want to see them grow up. I don't need to live for 30 years. I just want to be there at my daughter's 18th. I always get very emotional because she was 2. And when Melita said to me, you've got, 5 years, she's 2. I mean, I've got to be there for her. So I emailed him, and to his great credit, Professor Kipps said, I can see you next week. How's 2 o'clock, Friday? And I flew to LA, and I got the mad taxi driver down to take me down to UCSD in San Diego, and thought I was going to die on the road, and thought this is just ridiculous. And Tom Kipps said, look, I'm not going to, you've got all your medical records, you've got all your paperwork, I'm not going to repeat any tests. I'm just going to say to you, do not have a transplant. There are drugs, and ironically, one of the drugs that I think you should be on was invented in Melbourne. And I'm not sure why they haven't got you on this drug, but it's called ABT 199. Go home, go to your doctor and talk to them about this drug and find out if they can get it on a trial. Now, of course, my doctors did know about ABT 199, which is now venetoclax, but they couldn't get it for me. There was no trial open for me at the point that I really needed this drug. And the next day they had a patient conference, because in America, they actually recruit patients for clinical trials. So it's almost like an exercise where there is so much patient information and so much promotion about clinical trials. And I went to this conference, and by this stage, I'm barely awake, I'm jet lagged as hell. And I creep in the back and I sit down, and there's this Scottish doctor talking, and he's talking about, he's presenting, and I've actually got a photo of the first time I saw him. And I turned to the person next to me and I said, who's that? And I said, oh, it's Professor John Gribben from St Bartholomew's Hospital in London. I said, oh. And John, who's now a great friend, came down. He said he's never done this before. He came to the back of the auditorium and sat next to me. And he's normally, there's 300 people in this auditorium. And he said to me, don't judge me, I was wearing a koala on my jacket, because I was trying to connect with some other patients that I'd met on the forum. So I'm like, I'm going to be the one with the koala on the jacket, then you'll know me. And he said to me, wow, you've come a long way. And he said, are you a carer? Because you'd imagine someone, I was then 40 years old, you'd imagine someone was a carer, not a CLL patient. And I said, oh yeah, no, I'm a patient. And he said, but you haven't had treatment. I said, I've failed FCR. And he said, oh, okay, well, you're looking for a trial? And I said, yes. He said, have you still got your British passport? I said, yes. He said, okay, if you can get back to London in the next month, I can screen you for a clinical trial of ABT 199 and obinutuzumab. I actually think this is your best option to avoid transplant. Go back home, talk to your doctors. If they agree, I'll see you in London in a month's time. Well, I came back thinking, no way. By this time, I'm seeing people at Peter Mac, I'm seeing them at St Vincent's, I'm seeing, David Ritchie at the Royal Melbourne. No way am I doctor's going to say my best option is to leave Australia. And the last person I asked was Con Tam, who is now my main doctor at the Peter Mac. And Con said, oh, we've got some other trials that maybe that'll open for you. Maybe this will be open. I said, Con, what would you do if you were in my position? And he said, I'd be getting on that plane to London. It's a great trial. This is the only way you're guaranteed to get ABT 199. We know how well this will work for you. If it doesn't work, you're going to come straight back and you're going to have that bone marrow transplant. And we'll know that we've exhausted all options. So I remember going back to work, and I was working full time, and I just burst into tears. And I just thought, how can this be the answer? Leaving my children and moving back to London. But I did it. And I did that in July 2015. I couldn't get on the trial straight away. They delayed opening the trial. So I went back to work at the BBC with all my colleagues going, what the hell are you doing here? And I screened for the trial, got on the trial, because again, just because you screen for a trial doesn't mean you're going to. So I'd taken a lot of risk to get there. And I was the first one in England to have ABT 199, which became venetoclax. So as they're giving it to me, the trial had closed because some patients had died in America from tumor lysis syndrome, and this was the first trial to open after these they'd given overdoses, and it was 2 patients had died. I remember everyone being I knew one of the patients. We were all very, very scared that the drug would die as well, because we knew how powerful it was. And John Gribben said to me, Deb, don't worry, I've spoken to John Seymour at the Peter Mac, and we're following the Peter Mac protocol. So I'm sitting in a hospital bed in London being told, don't worry, you know, we're looking after you in the same way that Peter Mac would look after you. Within 5 weeks, I was the first one, which is actually the standard of care now, to have obinutuzumab to lower my tumour burden. And because I have bulky disease, I was high risk. So 5 weeks of obinutuzumab followed by venetoclax. And there's a photo of me holding the first 10 milligrams of venetoclax that I took, and I'm petrified. And, you know, flash forward 13 months and they allowed me to move back to Australia as long as I came back to London every month. So I commuted for 10 months in a row to London. Taking the drug for the first time, and then you flash forward and that had become part of my life. So I did very well on venetoclax. It got me into undetectable disease status, which is amazing because chemo didn't do that for me. And I stayed on it. I stayed on the whole of the trial. But in 2018, the trial closed. I was told I didn't have to commute to London anymore. And so I'd been on the trial for 2.5 years. And the blood test I did at the Peter Mac to collect my compassionate access to venetoclax was the one that showed it stopped working for me. And
Deb, I'm going to stop you there because I think people want to hear the rest of that story in a minute, but just to leave them hanging to see what happened next. But because I think we're sort of at the same time as where we're dealing with Janet in Melbourne with her problem. And so I'm going to let you rest and have a drink there, because I'm going to ask Barry, who also, very active life before the diagnosis. Some of you may recognise Barry from his TV appearances. You're a TV star, Barry. Tell us about how you got to where you are, and then about your journey with your plasma cell disease.
Thank you very much. And let me just say, if there's 380 people in this room, Janet and Deborah, your stories are just blowing us all away. You're incredibly inspiring women, and your sense of strength, and it's just so rewarding to listen to you speak. It's incredible. I want to say this, mine's going to sound like the TV commercial you watch when you're watching an amazing movie after those two pieces, but
I don't think so, Barry.
I'm a Channel 10 presenter. I'm on a Friday evening show called The Living Room. It's a lifestyle show. I believe it's a great way to end the week. It's very relaxing. I've got 3 amazing co-hosts, and we have a lot of fun, and we're very lucky to be invited into a whole bunch of lounge rooms every Friday night. But I didn't want to be on TV, that's for sure. I was very lucky to retire in 2005. I was 45 years old at the time. I'd been very successful in business. I had a great balance in life. I was a hard worker. I was very fit. I was a surfer and a sailor and an adventure man, if you will. And I had been retired for about 6 years. Yeah, 6 years. And it was around 2011. And my wife and I are very lucky to have a yacht in the Mediterranean. We spent 6 months of the year in the Mediterranean, 6 months of the year in Australia, in Bondi Beach. So I was a swimmer and a surfer, and for at the time, a 50-year-old, an incredibly healthy person. My wife in 2004 had suffered cervical cancer, so we'd lost the ability to have children naturally, and we were searching the world for an alternate for that. And for us, that was surrogacy. But long story short, a friend of mine had a lot of video footage of me on the yacht, and I've always been pretty confident sort of a character and always got plenty to say. And he's a TV person, and he showed another TV person, this guy on the yacht, and they decided that they wanted me to be in the lifestyle show, The Renovators, which is way back in Channel 10, quite a while back now. And they kept bringing me up and saying, hey, Barry, we'd really like to cast you for this show. It's an exciting new show. You're going to be the building expert. Your knowledge is incredible. And and you've got the right charisma for the TV. I said, I'm not interested in TV. I don't want to be in TV. I've got a great life. I'm very happy. So TV people don't give up though. They kept ringing, and I was back in Australia one Christmas, and I was down the South Coast surfing, and I was surfing along the side of the wave, for those that don't know how to surf, and the wave sort of curled over the top of me and hit me in the side of the head. With that, I was knocked off the board, but it was a very scary experience. My whole head went pins and needles. I could feel my body, so I knew I wasn't paralyzed, but I knew something was pretty bad, But me being a builder and an athlete, I played a lot of footy, and that I'd had a lot of knocks. I just rested. I thought the best answer here is just to rest. And that was at the Christmas time. And I was having massage therapy, acupuncture, all things like that from Christmas right through to March. And in March, we were going over to India for our, I think, 5th or 6th time at surrogacy. And I said to Leonie, my neck, any sort of jolt was just, the pain was agony. And I think I have a high pain threshold, but I just couldn't take it anymore. So I went to my local GP. Well, I didn't even have a local GP. I didn't go to doctors. I didn't like doctors, but I used to be a boxer a long time ago. So I rang up the old boxing doctor from the gym, and he said, come down and I'll x-ray your neck, and we'll work out what's wrong. When you get back from India, we'll get started on some therapy. So I go in and saw Jim. We talked the breeze, and it was all pretty casual. I didn't look unhealthy. I was not unhealthy. I was a very healthy human being. And he took the x-rays, and off I went to India, and we had a couple of weeks there, and I came back, and coming back, I said to Leonie, when I get back, I've got to get this neck fixed. Whatever it is, it has to be fixed. I'm getting dizzy. I was actually losing vision, my peripheral vision. I was losing that. And I'm an impatient guy, and I was an impatient guy. Cancer teaches you to be very patient, I'm afraid. But I had lined up to see the best physiotherapy at the Institute of Sport out at Homebush, and they said, I won't see you until I get the x-rays. So all I wanted to do was get the x-rays. I didn't really care about Jim's opinion. I just wanted the x-rays to take him out there. So I get in there, and as it was, he wasn't there on the day. And imagine this, I've ridden my Harley Davidson to the local clinic. And I said to the lady, I'm here to pick up my x-rays. I had them before I went away. They should be here. And she said, oh, that's all right. We'll wait for the next doctor. I said, I don't need to see a doctor. I just want my x-rays. And in hindsight, I think I was a little rude. And she said, and I said, no, really, I haven't got time to muck around here. I just want to get the x-rays. And she said, well, I'll tell you what, I'll read the report. If it's okay, you can take them. If not, you're going to have to see a doctor before we can release it. That's the protocol. And I'm not one to go to doctors. So I just said, okay, what the hell? Have a look, And it was really funny. Listening to Deborah about you were saying how you're a journalist and you saw things from a third person. From this moment on, and I say I'm a strong guy all the time, I start to tear up. From this moment on, I started to see life as the third person, because she lifted up the report, and she didn't even look at me in the face. She used a 4-letter word and said, you'll have to see a doctor. And I said, I got a sore neck, but it can't be that bad. And she said, she straight away picked up the phone. And I don't know who she called, but a doctor came straight out, looked at me. This is a person I've never met in my life. And doctors are beautiful humans. There's no doubt about that. But I'm in a room I've never been in, looking at someone who I've never met in my life, who's looking at a report, and she's got her hand on my knee and she's crying. And she says to me, Barry, do you have family? And I'm not a journalist, but my life seemed to be not my life all of a sudden. I was just watching something that couldn't be real. And I said, what's wrong? You'll be all right. What's wrong? You know, and I was trying to comfort her. And she said, Barry, it's not good. You have an enormous tumor in your neck. She said, I'm not sure if it's in your brain, but it's wrapping around the base of your brain. It's in your skull. She said, this is terrible. She didn't know what to say. And she was, like I said, physically, obviously very emotional and physically upset. And I was trying to calm her down, but I've never been sick in my life. And it didn't make any sense to me that I was, because I felt great. And she said, I think it's best if we call an ambulance and take you to the hospital. And I said, this doesn't make sense. I mean, what part of me is wrong? And I'm a popular character. I don't want to talk myself up, but I know a lot of people. So I jumped on the phone and I said to a friend who I know has a brother-in-law at St. Vincent's Hospital here in Sydney. And I said, Ronnie, what is it I've got here? I said to the doctor, and she said, you've this. And so I said, can you find out who's the best person? I've got to see someone. And she thinks I've got to see someone who can control the structure, because I haven't explained, but what has happened is my C1 vertebrae has been eaten by a plasmacytoma myeloma from the inside out. So it had eggshelled my C1 vertebrae. It had eggshelled part of my C2 vertebrae, and it was eggshelling my skull as well. So it was, and guys, doctor, and you may be able to explain this better than I, but the cancer was eating the marrow and the inside of my bone, and it eats it right out to the walls so that they're like a very frail eggshell. And that day when I was surfing, what had actually happened was I'd crushed the C1. We'd lost the connection between my spine and my head. So my head's just sort of floating in the breeze, and the only thing that was holding it was my muscles. That's why I had severe headaches. Those muscles were inflamed and they were crushing my arteries there. That's why I was losing peripheral vision. But the long story short now, I said, I'm not getting in an ambulance. I'll ride my bike home. And my mate has told me to see Tim Steele, who's the guy who can handle the structure there. And it was interesting because the first question I got asked too, have you got insurance? And I didn't have insurance. But this is where TV comes into it. I go into St. Vincent's Hospital, and they said to us, my wife met me in the hospital. I did go via ambulance, and he laid me down and he said, what I want to do, you've got something in there. We're not exactly sure what it is, and I want to biopsy it and try and work it out. So we have to put a needle through the side of my neck, and that was going to go past an artery into the C1. And we're going to draw something out and then try and work out what to do next.
Sounds easy.
Sounds easy. and it's really funny because I'm petrified of needles. And he said, I said, well, you knock me out, you do whatever you like. You know, put me unconscious and you just do whatever you do. Let's just get this fixed. I got to go and pick up my bike, you know. And so but to explain to the people watching what that means, it's a very difficult process because you've got to push this thing a long way in, and you've got to go past arteries, and you've got to make sure you've got it. Once you're there, you tap it. So they're continually sliding me in and out of a CT. Was it a CT X-ray? Just to make sure, and then they slightly adjust it. My head's turned, my wife's crying. Everybody's disappointed and everybody's upset, and I'm in this third person. I keep looking at you, Deborah. I'm in this third person sort of place, and after all that's happened, you know, sweating, doctors sweating, it was quite wild. But I just didn't see that was happening to me. I was just watching a bad movie, and it was really funny. I love to say it was funny. It was petrifying, but I don't invest in being scared. I don't see any advantage in it. But the long story short, that afternoon we had 3 doctors. Tim Steele said, I can manage the structure. I'm going to put steel rods here, there, and the other. The other doctor said, not sure yet. This is bigger, and you can't have had this for too long. The pain, blah, blah, blah. And the other doctor said, I would suggest you go home. You've got about 3 months to live. I said to her, I'm not interested in that opinion. And I said to the other two, I'm a collaborator. I'm a team guy. You do whatever you've got to do. I'm not going anywhere. I want everybody to know I hadn't had children yet. I had an amazing life. I wasn't scared of dying. I don't think I was anyway. Now, this is the funny thing, and this is what makes me smile about this story. I said, I didn't think I was going to die. I never think I'm going to die. But I said to myself, Baz, you've got property in Europe. You've got a boat that you love. You've got things that if I've only got 3 months to go, I'm going to have to sort some stuff out here. You know, it's as simple as that. So I turned on the phone to ring up my brother, and I was getting a little emotional too. And I think my wife was there. She was upset, but I wanted my brother with me. And you wouldn't believe it. There's a private number comes on there. And I don't know how many people in the audience have ever spoken to a casting agent, but they're really high energy people. Okay. And for some reason, you imagine the room is going in slow motion. There's doctors around there. Everybody's confused and the phone's ringing. And I answer it in front of everybody. It shows my own self-importance. But you hear this, I go, hi, Baz, it's Kirsty de Levant's Cast of Thousands. We are really, really keen to get you on this show, and we will do anything to get you on. And I looked at the phone and I thought, this can't be true. What is happening to me today can't be true. And I looked at the window. I thought, do I throw the phone out the window and just pretend and wake up or what? And I looked at the room. It's going to bring tears to my eyes because it was a crossover from realization, fear, self-belief, and just life. And I said to her, I'll tell you what, you ring me back in 3 months, and if I answer, I'm on. And I shut the phone. You're not going to believe it. 3 months later, I'm walking out of St. Vincent's. I've got all these plates in my neck, and I've got this bracket holding my head on, and I've had, I think, 9 or 10 weeks of intense radiotherapy. My head was like a microwave, you know. And I'm walking out the door, and remember this, I'm this big, healthy guy. He's 115 kilos, pretty muscly, you know, when they were saying they wanted me on TV. She rings up on the day 3 months later and says, Baz, you said if I rang you back in 3 months, you'll be on the show. Well, I weigh now 78 kilos. I look like I got HIV and I can't move my head. I said, yep, when do you want me? She says, can you come in on Thursday? And it was like a Tuesday. I said, I'll be there. And I was literally wearing pajamas for 3 months. So I had to go and buy a new outfit and put a new outfit on with switches because all my clothes were too big for me. And I walked down the driveway of this set where we were going to do the casting. And they said, you've lost a bit of weight. and I said to the guy, I've been through some stuff, but if you want me, I'm here. You wouldn't believe it. I think it was only 4 months after that I was on TV, and I was the host of the Renovators, and I was healthy up until about 2018. But that's another story.
So we'll come back to that. So we've got, I mean, clearly it's, you know, the issue of you know, the shock of being told that you've got incurable cancers, and you're all here. So with the treatments that we have for the conditions, Janet's had precision therapy, new drugs. Deborah's had the new drugs, but also has had CAR T cells. And we'll talk about that. So Deborah, I really need you to fast forward. And you've had CAR T cell therapy because the venetoclax eventually ran out of steam. Other treatments ran out of steam. You had CAR T cell therapy. When was that?
September last year. So in the middle of COVID, my T cells were harvested using that machine, which is quite a brutal process, and amazing nurses, you know, skimming off the T cells, and that takes 2 to 3 hours. And then they were sent to America for manufacture. And they were delivered back to me. It took 8 weeks to deliver them back because of COVID. And so I had to go through screening twice for the same trial. Barely made it onto trial because ibrutinib that I was on, the BTKI inhibitor, had done such a brilliant job of bringing me back into remission. And then they gave me 3 days of chemo before they gave me the T cells. So they said to me, there's a good chance these won't work for you. You know, they need disease to fight. But Barry's saying, you know, sorting stuff out. It's the first time in my then 9-year journey that I did an advanced care directive. I did a will. I did executive power of attorney. I sorted everything out. I was convinced that this was bloody dangerous. It really was dangerous what I was doing. I used a new CAR-T product, which again, and when it was reinfused, Michael Dickinson and Con Tam were my doctors at the Peter Mac, and they'd said to me, look, just watch out for day 11. You're number, I think I was number 3 in the world for this brand new CAR-T product with CLL. And they said, we'll keep you in hospital for 5 days, but we're not expecting anything to happen. And nothing did. I just watched a lot of Netflix and updated my blog. And then day 11, I was actually watching the Geelong football match, and I was starting to feel just a bit shivery and cold. And my partner, who, you know, had to really step up. I mean, he's my boyfriend, and he suddenly had to become, you know, my medical carer. And my mum couldn't fly down from Queensland because of COVID. And he said, you're not looking great. And I said, I'm feeling really cold, but I'm sure I'm fine, but let's just watch the end of the footy. And he got a thermometer, and I was burning up, but I was cold to touch.
And this was your T cells.
This was the T cells. And an hour earlier, I'd actually emailed Con and Michael going, what happens if it hasn't worked? Can you like put some more in, or, you know, what happens? Anyway, they bloody well worked, because it was suddenly, I was, you know, in the Royal Melbourne, I was in a high dependency unit, I'm having 20-minute obs, I am so sick, it's like every part of me is having a bone marrow biopsy at the same time, I'm in agonizing pain, and the only thing that's stopping the pain is Panadol. How good's Panadol when you're in pain? I had no idea. Just keep giving me the Panadol. They didn't want to give me anything other than fluids because they didn't want to dampen down the response. But I remember saying to Marianne Anderson after she came from the Peter Mac in the morning, and never have I been so grateful to see people from the Peter Mac, because it was pretty miserable in the Royal Melbourne that night. And I said to her, how long is the pain going to last? What's the data saying? And she said, Deb, you're number 3 in the world. You are the data. You've been incredibly brave. I'm like, I don't want to hear this. I just want you to tell me that it's going to stop. And because it was a new product, and Sam, you know, you can probably help me with this, but they'd engineered it in a different way from the ones that have been FDA approved. So I was already scared witless about the fact, but this was my only chance to get CAR-T, and it was eligible for the trial. I'd missed out on trials before. I didn't want to have to go to the US when, you know, and if I didn't do this, when ibrutinib runs out of steam, which they told me it would, when I fail another novel therapy, I'm in a lot of trouble. So I signed the paperwork and did it. And I'm now a year this year. Do you want me to tell the punchline?
We'll come back to that, because we'll finish it off there in a sec. So Sam, these CAR T cells. So Sam, you're an expert in CAR T cells. So briefly, in a minute, tell us why you're here in Australia, what you're doing in America, and then we'll talk a little bit about CARs.
So thanks Miles, and again thanks Deborah and Barry and Janet. They're all just amazing stories. So I'm a postdoctoral research fellow, so I've done my PhD. I did my PhD with Ian Fraser, inventor of cervical cancer vaccine, back during my medical degree, and then I was told that T-cell therapy is never going to be a thing after my PhD, and I should just give up on it. And I did, and I went into hematology and did hematology training, and then all of a sudden CAR T-cells started coming back on, and I thought, well, I've got to get on this, right? Because I've done T-cells already, and I've got to get back in the T-cell world. So I was finishing up my hematology training at the Royal Melbourne and Peter Mac. And at which point I turned to Dave Ritchie, who you mentioned before, and I said, Dave, I don't want to do any more training. I'm going to America, right? I've got to get on this thing. I've got to understand what's going on with CAR T-cell therapy. So I received a scholarship from the Hematology Society of Australia and New Zealand, which I'm very thankful for, and went to Seattle to the Fred Hutchinson Cancer Research Centre, where I got a postdoctoral position with Cameron Turtle, who is one of the world leaders in CAR T-cell therapy and is a Sydney expat as well. and a fantastic clinician and an amazing researcher.
So we've touched on a few names here. It's interesting that Marianne Anderson is actually a Snowdome, is the Gandel Walter and Eliza Snowdome Fellow, which we've supported for her work to continue the work on venetoclax and how it works. It's interesting that, Sam, we support the Leukemia Lymphoma Society of America. There's a fellowship, and in fact, that's just been announced. And Kath Cummins has just come back from the other side of America, over at University of Pennsylvania, and she's just become the a new fellow, a Snowdome fellow, as appointed. So I won't tell you all about it. People should go to the website as well as check the Snowdome feeds. But she is just been appointed and she's a CAR T expert like you, and we want to get you back to Australia, and that's happening. So currently now you're a postdoc, you're running a lab in America while you're working here during COVID, and tell us about what the passion is with CAR T cells. What is going to happen with CAR T cells in other disease? We've talked about CLL. I talked about leukemia, lymphoma. Barry's got myeloma. Where are we going to see CAR T cells in reality in the next 2 to 3 years?
So I definitely think myeloma is the next big one, because it already is a big one. So we're already seeing ide-cel it's approved by the FDA in America, showed some outstanding responses in a trial.
What was the response rate?
I believe about 86% overall response rate, and
in patients that had had a lot of treatment.
I think median 5 lines of therapy prior to CAR T cell therapy. So these are very, very overtreated patients. And we know that myeloma wrecks your immune system. So we can only imagine the activity of these CAR T cells taken from patients if they're taken earlier on in their treatment course. So I think the field of myeloma is incredibly exciting as far as CAR T cell therapy and the capacity to improve myeloma therapy. And
so
then
it works in chronic lymphocytic, the main, what are the big guns, and what are the second ones? What's happening next?
So the disease that probably has the best response out of everything to CAR T cell therapy is acute lymphoblastic leukemia, or ALL, which is a much more common leukemia in children, but still does occur in adults.
And that's available in Australia?
That is available in Australia. So that's a product called Kymriah that's approved by the PBS up to the age of 25. And then non-Hodgkin lymphoma, or diffuse large B-cell lymphoma, which is the cousin of the T-cell lymphoma that Janet mentioned before, and it's got impressive activity there, about 50 to 60% complete response rates, and about some 70% sort of including partial response rates. And then myeloma that we mentioned as well, and then the disease that I work on, which is probably the most aggressive fatal hematologic malignancy of adults, and that's acute myeloid leukemia, or AML.
What's different about that, and what's challenging about acute myeloid leukemia?
AML is a particularly challenging disease to treat with CAR T cell therapy on multiple fronts, but probably the most challenging is the fact that CAR T cells have to be directed against something, and that something is usually on the tumor, right? So in the case of CLL, it's directed against a protein on the tumor that's on B cells as well. Now, your CAR T cells go and kill off the CLL, but it also kills the B cells. You can live without your B cells. We can replace the protein that your B cells make by giving you infusions. And so people can live without them. But the problem with AML is the proteins on the surface of AML are the exact same proteins that are on our stem cells in our bone marrow. So you wipe those out, and then all of a sudden you don't have any bone marrow capacity. So it's truly throwing the baby out with the bathwater there. So this is the challenge of AML, and this is the challenge that we all face. And we've got ways around it. But the other
don't sound like you're too afraid of a challenge, though, Sam.
No, not at all. And I think we've got some really exciting data, actually. We've got some really exciting data in it.
So don't let Sam hide his light under a bushel. His work is really at the front end. It's the top, 0.1%. He is doing some amazing stuff in acute leukemias and generally with CAR Ts. I showed a slide, and everybody asks, and there's questions up there. Why is it so expensive? Is it always going to be expensive? Is it going to be affordable? What's your thoughts?
Yeah, so in fact, this is the first thing I did when I landed in Seattle, was write a review on affordability and value of CAR T-cell therapy, because I was just so interested in it from just a practical perspective, right? Australians are practical people, and so the thing that's really important to remember is that when we talk about cost, you've got to talk about cost effectiveness or cost efficacy. It's an expensive drug, but it's an incredibly effective drug. And with time, will not only improve the cost as it gets cheaper to manufacture, and these things are expensive to manufacture. You've got to take the T cells out of a patient. You've got to introduce this virus that you mentioned that's very expensive. These things cost a fortune. And then you've got to grow these things up with those herbs and spices that are also very pricey. All of this is done in a lab with 2 people standing there, one with a checkboard, checking them as they go, bit by bit, every little milliliter by milliliter as they put it into a dish, right? So it's an incredibly manual process at the moment. So hence it's so expensive. As we start to automate the process using sort of closed loop manufacturing systems, of which there are apparently about 3 at the moment, it'll get cheaper. In fact, there's some work in Sydney showing that you can get the cost down to about $20,000 Australian for a single infusion of CAR T cells. And that'll make it cheaper, but also understanding improving their effectiveness will clearly make them more valuable, right? So it's 2 things that are certainly going to happen with time, and new papers get released every day, of which there are 1,000 current trials in the works.
Sam, sorry to jump in, Miles. One of the things that I was really interested in was the research about whether CAR T will work for a certain patient. That there's new data about being able to test whether the CAR T will work before patients like us who might not be able to get CAR T here but have to come all the way to the US. Have you seen that research? What would you say about that?
Yeah, absolutely. So I guess there's, traditionally in medicine, we understand and we can sort of attempt to predict how patients are going to respond based upon that patient themselves, right? So we know with CAR T-cell therapy, if you've got too much disease, you can't cure it. If you've got too little disease, like you mentioned before, or no disease at all, it's difficult to get those things to work. So we know the clinical factors that determine responses to CAR T-cell therapies, but we're starting to understand the genetic factors that determine those responses. And that's actually part of the work I work on as well. And there's some also interesting work understanding the gut microbiome. So your own bacteria in your gut affect the activity of these CAR T-cell therapy, and optimizing those can also have a function in improving outcomes for these therapies.
So, I mean, it's amazing how we have to learn to be synergistic with our immune system and work with our bodies and start working with our bodies to fight the cancer. So we're basically running out of time, but there's a few questions here, so I'm going to go through them relatively quickly. I think the first thing to say is mantle cell lymphoma. It's on the list of CAR T cells. There's questions about myelodysplasia, which is actually close to acute myeloid leukemia. And I think as Sam cures acute myeloid leukemia, watch this space. Then myelodysplasia, a big group of diseases. Just to be clear to people, what Deborah was talking about was an allogeneic transplant where she gets it from somebody else, which is the ultimate immune therapy, where you're getting an immune system from somebody else, but can be very toxic, and as opposed to autologous, which is much more easily and acceptable treatment. So I just wanted to clarify that. But now I'm going to finish off. I am going to say a couple of words, firstly, to thank all of you. But I wanted to, in all of these discussions, it's all been about time, buying time. Sam is the Time Lord. He is the man, and people like him, who are the researchers who Snowdome supports and who you support by supporting Snowdome, are the Time Lords who are making discoveries. And we've had many of these on our various panels, and we've all learned what makes these people get up and drive and want to cure, because sadly we do have to break the news. And I think Barry's taught us about what it's about, like breaking news, and what's it like to hear it and how it impacts on your life, but also the fact that we can look at him now. We're going to get you back and hear your full story, Barry, except to say that your disease is doing well. I'm going to ask each of the panelists, Janet, Barry, and Deborah, to just say, Janet, you've bought some time. What has been the most important thing that time has brought you?
I guess the most important is the birth of my first grandson.
Yeah.
I never thought I'd live to see that.
When was that?
He's 7 weeks. And because of COVID, it's hard, but I've seen him twice, and that brings such joy. And when I was thinking about tears, blood, sweat, and tears, all the family is worth it. But he's been, so I've been lucky to see him.
Yes, blood, sweat, and tears. Barry, what's time brought you?
Well, I mean, I now have multiple myeloma. We're managing that really well. But since the plasmacytoid myeloma, I've got my beautiful twins, Bennett and Arabella, who are my life. And time is a precious thing, and what you people are doing is incredible. And yeah, it's brought me life. Life is the most precious thing there is, and it's very special.
Deborah?
Well, my children are now 17. Oh my gosh, it's so exciting. 17, 14 and 11. So it's brought me an extra 5 years with them that I didn't think I'd have. And time has brought me a total change of direction. So I now feel very strongly about making sure that no one has to go to the lengths I've gone to get the best treatment. No one has to go overseas for treatment. We get people like Sam to come back to Australia. And thank you to everyone who supports Snowdome, because you're making a real difference to people like me and to our children and our grandchildren and our families. So thank you, and thank you for this evening. It's been amazing. I'm quite emotional about hearing everyone's story. And from my point of view, I'm a year post CAR T now, and I have no detectable disease, and I've been off all drugs other than alcohol since March. And I'm just incredibly grateful to all the scientists and the doctors who've got me here. So CAR T is the future, and there may be other treatments, but I'm very grateful to have been able to have this at home, and that's thanks to you, Miles, as well.
So I just want to finish off, and I think there's been some important questions and comments in the last few minutes which has come through, which is that talking about giving these sorts of new treatments earlier. And I think this is the focus of time as well. What we've learnt from the group, from all of you here tonight, is the bravery. And I think, Deb, the explaining of being so early in the treatment. Janet, having the acceptance of the recommendations and saying, I'll try this therapy when all seem to be lost. Barry telling the person that I want to live, and I'm not interested in that process. I want some time. And these new therapies are going to come earlier, and we've already seen that, and Sam's touched on it. And I think we will find that as treatments move earlier, they'll have more impact, and we will move to a day without chemotherapy. But it can't be done without this support and investment. And through Snowdome, basically, our view is we fund people like Sam, largely, and it's about supporting people who are smart, who can do the trials, track the companies to say, let's do the trials here in Australia. And but unfortunately, it's not just about money, and we don't want to compete with pharma. We want to be able to say, we can provide people who can do the trials. And we've done that in Western Australia, which have gone from virtually nothing to almost 100 trials. Snowdome's raised $40 million in the last few years. It takes a lot of money, but it is supporting people. We've supported numerous people, and because it is about getting the rubber to the road, so we can write clinical trials, be attractive to drug companies, get the drugs and treatments over here. And CAR T cells is a big one. It's expensive, but I can tell you that next year we've got trials that will be done the first in the world for CAR T cells. So it isn't like Australia's behind, it's just have to have the resources, and it does take a lot of money, but it's incredibly inspiring to hear that what we've heard tonight has happened in the last decade. These changes have happened in such a short period of time. So we're fighting time. And as you know, as I often say, Einstein had a equation which is E equals mc squared, which was about the speed of light and time. Well, I think the dollar sign can go in that, because my point of view is that the only thing that can speed up research is money. And so I will finish with a thank you to the audience for all of you joining. I can see we've hung on to everybody who joined us. It's because of the inspiration that Barry explained, and the inspiration from Barry and Janet and Deborah, the inspiration we get from Sam. I'd like to thank each of you individually. I'd like to thank the Snowdome people who have put this together. And I'll finish with an unashamed request that this is Blood Cancer Awareness Month. And we, unfortunately, nothing we know can cause or prevent blood cancers. You heard the randomness about it from all of our guests. And so please go to the Snowdome website, it's snowdome.org. You can kiss away a vice and try and raise some funds for Snowdome, please do, or you can just leave us a big wad of cash, and so that we can find the next Sams and bring them back to Australia. And I've got a list of here in the chats that say, bring Sam back. And we all want to do that. But Sam is one of many of the amazing talent that we have here. So good night to everybody. Thank you for joining us. You'll get to meet Deb and Barry and Janet again. And thanks, Sam, and thanks everybody from Snowdome. And everybody, have a wonderful evening.
Thank you very much.
Thank you.
Thanks, Miles, and thank you, Snowdome.
Bye.